Currently, I'm a student. I'm a PhD student of the Charles University in Czech Republic, and I'm doing my PhD in the Laboratory of Ligand Engineering of the Institute of Biotechnology of National Academy of Science of Czech Republic. And so I would like to say that I'm glad to present here today and to be able to see my teachers, and I would like to introduce you a project I'm working at. The aim of this project is to create small protein scaffold binders to inhibit interleukin-6 cytokine. So interleukin-6 is a pleiotropic cytokine which has diverse functions in human organism. It functions through the heterodimeric receptor, which is composed of alpha and beta subunit. So at the first step, interleukin-6 binds to its alpha subunit, and this complex assembles with the beta subunit, and then I'm sorry, if everything okay with the sound? Я перепрошую, мене добре чути чи ні? Я Кирило Миколайович, мене добре чути чи ні?
Добре чути тебе, але це зупинилась презентація чомусь.
Зараз видно?
Нормально, все нормально.
So I will continue. So interleukin-6 binds its alpha receptor, and then this complex assembles with beta receptor, and then two of such trimers assemble into the hexamer, the functional unit. And alpha receptor is required for complex assembly, while beta receptor is able to transfer signal of the cytokine through different pathways like JAK-STAT and MAPK. So the trick is that beta receptor is present on almost all cells of the organism, while alpha receptor is present only on some of them, but this cytokine has different modes of action, and that is for it's able to spread its function on different types of cells. So it can bind through classical signaling pathway when both receptor subunits are present on cell surface, or it can bind to alpha receptor that is soluble, and in the composition of this complex, it can activate cells that contain only beta receptor but not alpha. And the third way of signaling is transpresentation of interleukin-6 by dendritic cells to CD+4 T cells. So, and in this way, interleukin-6 is able to function on many cells, and mostly it's known about because of its function in immune system. It's important for immunoglobulin production, for CD for T cells survival and differentiation, and for the adaptive immunity. But beyond immune function, immune system, it also has a lot of other activities. So it's important for liver regeneration. It participates in neural inflammation, in blood vessel angiogenesis. It's important for one killing. Takes part in bone homeostasis, and it's also participates glucose and lipid metabolism. So, and as we can see, it's a very important molecule in the organism, and some dysregulations in its production can lead to many pathological states, which include autoimmune diseases, cancer, and also it was shown that interleukin-6 participates in cytokine release syndrome during COVID infection. So currently we are focused on the function of Interleukin-6 in AGA nephropathy. It's a kidney disease, and it occurs because AGA antibodies produced in the body with a wrong glycosylation pattern. And this wrongly glycosylation part of the AGA is recognized by autoantibodies. And as antibodies are huge molecules, they are able to form huge complexes that fail to infiltrate through kidney. And they injure kidney, and in some cases it can cause renal failure. So during this project we want to, and it was shown that interleukin-6 plays role in these aberrantly glycosylated AGA antibodies production. So we would like to create inhibitors to block interleukin-6 and to see if we can use it as a therapy for AGA nephropathy. So it's not the first it's not the first attempt to create interleukin-6 inhibitors, and actually some of them are already approved for the market. So there are several strategies. So it's possible to bind to block interleukin-6 molecule itself, to block either of its receptors, or to block downstream signaling. So as we can see, most of the interleukin-6 inhibitors are presented by antibodies, which are great molecules, but they have their own limitations. And also there are some small organic compounds, and there is one interesting binder, which is interleukin-6 super antagonist, which is the interleukin-6 molecule that is mutated in that way that it can bind a receptor, but it's not able to transduce signal. And also one interesting representative is Avimer. It's a small protein scaffold binder, which is able to bind interleukin-6 molecule in 3 places. And this avidity effect ensures strong binding. And this molecule was actually investigated in clinical trials, phase 1, but its result was never published. So we would like to try alternative approach to antibodies to inhibit interleukin-6 by using small protein scaffolds. So as I told, antibodies are great molecules that were evolutionarily developed by our organism, but they are big and they have complex structure and they need post-translational modifications, and they have to be produced in mammalian cells, and so they are actually very expensive. So there is alternative approach to antibodies. It's a scaffold, small protein scaffold. So it's possible to take small protein scaffold to decide which amino acids of this scaffold are important for the fold and structure of this molecule, and to make them consider them as a conserved, and to mutate the other amino acids which are not important for the fold. And in such case, it's possible to create a huge library of protein variants, and among these variants, there is a huge possibility that there will be one which can bind your specific target with high affinity. So this approach has a lot of advantages because these protein scaffolds are mostly human, so immunogenicity is expected to be low, and also they are simple in structure. They can be produced in E. coli, and they are small, so they can have better tissue penetration, and also it's easy to manipulate with them, to conjugate them with some other probes or some other molecules, or to create bivalent binders, and they are selected in vitro, so it's possible. Immunization is not required for selection of such binders, and also the library of these small protein scaffold variants can be created once and used for many targets. So you don't need to create a new library for each target you want to bind. So in our case, in our laboratory, we have two types of libraries. So one is based on this albumin binding domain, the Staphylococcus aureus. So this particular protein is called Affibody, but we took the same scaffold and created our own library design. And also we have a scaffold that we created ourselves. It's a domain, or it's called myomedin, and it's a domain of the human protein myomedin. And now it's undergoing patenting procedures. So we want to use these both or one of these libraries to generate small protein scaffold inhibitor and to test it in the laboratory, and then to test it on the cells from the patients with IgA nephropathy. And so we hope that we'll have some promising results, which I will be able to present in the next conference. So thank you for attention.